Welcome back, everybody. So today we're going to dive into digestive function by looking at structure-function relationships in the early digestive pathways, in the oral cavity, in the alimentary canal, up to the stomach. In my last lecture, I discussed the four processes that drive digestive system function, specifically getting nutrients from a macroscopic state into microscopic states and ultimately into the body. So as a reminder, these processes are that food needs to be transported, specifically through the alimentary canal or the GI tract, all the way through. And anything that is in this tube is technically outside of the body, despite it being surrounded by the body proper. The goal of moving the food through the alimentary canal is to absorb the nutrients that the body needs and uses to support life. And in order to do that, to absorb those, they need to be microscopic, which requires mechanically breaking down, in this case, denaturing proteins, and then chemically digesting them via the action of enzymes and breaking atomic bonds until we end up with monomers that can be absorbed into through the digestive walls. Although we already talked about this in my previous lecture, I wanted to just review and be a little more explicit about the structure of different macromolecules that are being broken down and ultimately absorbed, and also review some terminology. So as a reminder, a monomer is a single molecule. So if we think of a protein or a carbohydrate, that's going to be the smallest molecule that is still classified as that type of thing. So here we can see our hexagon right here, which serves as a monomer. In this case, we're imagining it's a protein. So the blue is the amino acid lysine, green is the amino acid alanine, and the orange is the amino acid glutamate. So here we have a monomer of lysine, which is one building block of a protein. And so that's the smallest component that we can have that still is classified as kind of an amino acid or a protein. As these building blocks are bound together via atomic bonds, represented with these solid lines, we get more complicated structures. And so if we have two bound together, two monomers, we get a dimer, and three would give us an oligomer. Or we can also think about these, in this case, I'm using proteins as an example. Remember, you can also think about this in terms of sugars. So carbohydrates would have, the monomer would be a monosaccharide, and a disaccharide or an oligosaccharide would also be appropriate in the case of a carbohydrate. As the chain gets longer, eventually it turns into a polymer, which is multiple components, small components or building blocks, in this case of amino acids, all bound together. And as a reminder, in proteins, amino acids bind end to end, N terminus to C terminus, and so it ends up with a linear chain of amino acids. And again, all of these amino acids are bound to their nearest neighbors, forming this large chain. Finally, for proteins, we can think about how they fold, which gives it three-dimensional structure. And this three-dimensional structure is often critical and important for the protein's function. So in this case, I'm showing this protein curling up, and specifically it's forming hydrogen bonds. As a reminder, hydrogen bonds are attractions between a positive and negative charge within the molecules themselves. Although it's not strong enough that it's an atomic bond, these bonds are relatively easily broken, but they're still relatively strong. And just as a reminder of an example of a hydrogen bond that we talked about previously in the respiratory system would be the hydrogen bonds between water molecules that produce surface tension. So that surface tension can be strong enough to hold things up, like in the case of a water spider or a water strider. But they can also are not so strong that they require special or a lot of energy to break, or special situations or catalysts, like in the case of chemical or atomic bonds. So when we think of digestive processing, we think of two things that need to occur, and they're slightly different. So the first is that we need to mechanically break down substances. And as a reminder, this mechanical breakdown or mechanical digestion is the breaking of non-atomic bonds. So whatever forces are adhering this molecule, which aren't actually sharing of electrons that form those atomic bonds. So often this will involve breaking hydrogen bonds in the case of a protein, or we can think of non-polar interactions, like in the case of a lipid bilayer, where the non-polar polar tails are all coming together, and because they're all avoiding charged substances or water, they effectively adhere together. So examples of mechanical breakdown are mastication or chewing, denaturing a protein. In this case, that means unfolding the protein. So here we break this hydrogen bond and we can unfold and return this to a linear chain. And as a reminder, that increases the surface area of our protein to interact with whatever fluid it's suspended in, or emulsification in the case of lipids, which is effectively breaking them apart and mixing them together by creating other nonpolar interactions that don't result in them clumping together. The other type of digestion is chemical digestion, which involves actual breaking of atomic bonds. And so here we go from our linear chain into smaller pieces. This occurs usually or almost always via the action of enzymes. As a reminder, enzymes are proteins, so they have this three-dimensional structure which allows them to facilitate these reactions. And these enzymes are usually pretty targeted. So in this case, if we look at our protein right here, we can imagine we have two enzymes, this blue one and this orange one, and they will be targeted towards specific bonds. For instance, this blue one only cleaves bonds between alanine and lysine amino acids, whereas enzyme 2 only breaks glutamate bonds. So we can see in our action right here that this site, this alanine lysine bond, and this glutamate bond here were both broken, which gives us an polymer right here, a smaller protein, but it's still a polymer, and two dimers of amino acids. And so just note that this is very different from mechanical breakdown, right? If you're chewing something, it's very general, whereas the enzymes are very specific. So you can't use whatever enzyme you want to break anything down. There's not a universal enzyme. Whereas mechanical breakdown, you can chew all you want and it will effectively break down or separate, physically separate all different types of macronutrients, whether they're carbohydrates or lipids or proteins. And then ultimately, the goal is to use these enzymes to chop up this protein into the individual amino acids, which can then be absorbed into the body and into the bloodstream. And usually your amino acids have to be monomers in order to be absorbed because they need to be small enough to fit through the plasma membrane of cells. Digestion is occurring in the alimentary canal or the GI tract, the continuous tube that carries food through the body in this kind of cavity, internal cavity that is continuous with the external environment. And one of the things I want to note is that when we're thinking about the structure-function relationships, it's useful to note that the general structure of our alimentary canal is the same throughout this whole tube. So the general structure is the same, but there is functional variation in the structure as we go through here. And in fact, the devil is in the details, understanding what processes or how different parts of the GI tract achieve their function, whether it's absorption in orange or mechanical breakdown in this magenta color or chemical digestion in green, is really key to understanding how digestive processing occurs. And so some of the differences are relatively subtle, but they're often significant. So here we have a section of our alimentary canal, and you can see that this section has multiple layers. And we'll start with this inner layer. So we have the inside of the tube, which is termed the lumen, and our inner layer, which is the mucosa, which is composed of an inner epithelium, which is lining the lumen. And then this connective tissue layer, remember all epithelial layers have a supporting connective tissue layer. This is termed the lamina propria. Note that we have lots of blood vessels in here. So these are capillaries and lymphatic vessels where nutrients that are absorbed will go into the bloodstream and then ultimately to the liver and to the rest of the body. And then finally, we have this very thin muscularis mucosal layer. And so this muscle layer is very thin and it actually serves to kind of squeeze secretions out of glands. So you can see these little glands right here. And so these glands are producing secretions, and this inner muscular layer of the mucosa helps generate enough force that it kind of pushes the secretions into the internal cavity. But this inner muscular layer is not actually important for motility or transport. So let me give you an example of some of the functional variations that occur in this tube, and we'll dive deeper into this in the next lectures. So if we look for instance, at the epithelial layer of our submucosa, so this is the layer that's actually interacting with the contents of the tube, we can compare the structure of the stomach lining right here versus the intestinal lining. So note that they're both epithelial layers, but in the stomach, we see this pit structure. So we see kind of this large surface. This is all epithelial cell. And in that surface, there are these deep pits that go down where there are glands that release the gastric juices into the stomach cavity. And so this reflects the fact that the stomach is not involved in absorption. Rather, it's involved mainly in chemical breakdown, or mechanical breakdown, but also a little bit of chemical breakdown. Whereas when we look at the intestine, we see these villi. So these are fleshy projections, which are tissue level. And then if we zoom in even more, we can see on the cells themselves, there are microvilli. And as a reminder, these are both increasing surface area, which increases interaction. And so that interaction allows for more absorption. So the stomach glands are more protected, which kind of fits with this idea that inside of the stomach is very acidic. It's not actually absorbing stuff, so it doesn't have to have projections out into the stomach. Rather, the delicate tissues of these glands can be protected, whereas the intestines really want to interact and get dirty and pull whatever they can that the body needs out of the lumen and into the bloodstream. And you can see that here in this diagram on the right. If we look at our stomach, we can see there's mechanical breakdown, there's transport, and there's a little bit of chemical digestion, which we'll talk about later in this lecture. The next layer is the submucosa. So the submucosa is really important for both absorption and also chemical digestion. Note that it has glands present in it. It also has blood vessels and a huge nerve plexus, which is termed the enteric plexus. And so one of the things I want to mention is that the submucosa contains the enteric nervous system, which is these network of nerve fibers, and you can see what they look like here. So this is a fluorescent stain that's staining neurons or a neuronal marker. And you can see this star right here. This is actually our mesentery right here. And so you can see these vessels going down and our nerves coming in and innervating this section of the intestine and providing all this nervous innervation to activate different things. And one of the things I want to mention about this enteric plexus, this network of nerve fibers, is that it can largely work and largely does work independent of the CNS or even the autonomic nervous system in general. So if you think about like the sympathetic chain, and it does that via these short reflexes. As a reminder, a short reflex is when you have a sensation, say stretching of the gut here, which will be sensed by the neuron, and that neuron sends a signal to a ganglion, which is a local cluster of neuron cell bodies. And so you can see the ganglia here present in our submucosa, and then these ganglia directly send outputs to the muscles and generate responses. And so these muscles could be in the mucosal layer, triggering secretions from the glands, which are building up enzymes or other things inside the gland itself, or it could trigger activation of the next layer, the muscle, which actually causes things to move through the gut. The way the enteric nervous system works is that it has, or these cells release lots of hormones, neuropeptides, paracrines, and autocrines, which all interact with the nerve plexus and the local environment. So as a reminder, the gut is composed of smooth muscle. And so the smooth muscle is innervated by varicosities. And these varicosities are they're not synapses, they're kind of release the neurotransmitter or the neuropeptide very generally around the smooth muscle proper. And so it's not as targeted as normal muscle. And there's lots of different things that are released by the enteric nervous system that regulate secretion, for instance, regulates acid secretion or secretion of enzymes. And just some of these, I'm not going to be exhaustive, are gastrins, which include CCK, and I'm not going to pronounce it right now, secretins and somatostatins. And here on the right, you can see examples of these different compounds, where their target organ is and what their action is. And I've also kind of color-coded it so you can see a lot of these are involved in transport, which fits with their activation of muscle, but some of them are involved in mechanical breakdown, and some of them are even involved of actually pushing things out through glands and releasing secretions, for instance, for chemical digestion. So this brings us to the next layer, which is the muscularis, which is composed of smooth muscle. This smooth muscle is innervated by these nerves right here. And you can see that there's two layers. There's a circular muscle layer and a longitudinal muscle layer. So one going around the tube and one going along the length of the tube. As these hormones and neuropeptides and peregrines are released, they'll regulate the muscle activity and synchronize the motility of this muscularis layer, and it will trigger different movements. And so here I've shown two examples of movements. So one is peristalsis, which we've talked about already, which is involved in transport. So it's this propagating wave of contraction, which pushes a food bolus or chyme or whatever is in the intestine at that part of the GI tract further along the tract. The other example of a synchronized activity is segmentation, which is actually more of a mechanical breakdown, hence the magenta color coding. So these are alternating waves of constriction. So you can see multiple parts of the gut are contracting at once, which segments or separates the food in the gut. And then these alternate back and forth. And so what you have is mixing and interaction. So it kind of squeezes and crushes the insides of the gut and also mixes things together. So you can see these blue and yellow eventually mix together to form green. The final two layers aren't so interesting for our discussion about the function of the digestive system. So the outermost layer is the serosa, which is a serous layer which releases lubricating secretions to prevent damage to the gut tissue and also provides mechanical support. And then we also have these mesenteries, which are effectively a membrane sandwich, which contains blood vessels, nerves, and lymphatic vessels. And these mesenteries exist because it allows the body to more efficiently route these vessels and nerves to different parts of the gut. So rather than running along the length of the tube, which would be really slow and inefficient and probably wouldn't work very well, instead the vessels are sent down and they kind of branch out from these mesenteries. So it's much more efficient and provides a shorter path for communication, whether that's communication of nutrients or waste back to the body or communication via electrical signals through the nerves. So now that we've gone over the general organization of the GI tract, let's talk about the functions along different parts of the GI tract and dive a little into the structures that compose the digestive system and how they work and how they support digestive processing. And so we're really going to focus on these early pathways from the oral cavity all the way to the stomach. And then the next lecture, we'll look at the intestines and their role in digestive processing. So if we look at our early GI tract, we can divide it into different regions. We have the oral cavity, and you can see the oral cavity is involved in propulsion, chemical digestion, and mechanical breakdown. We have the pharynx and esophagus, which is really specialized for transporting the food further down the GI tract. And then we have the stomach, which is also involved in transport, a little bit of chemical digestion, mechanical breakdown, and then a very, very tiny role in absorption. So really our early GI tract is not involved in absorbing nutrients at all. It's really these three, either transport or processing and preparing nutrients for absorption, which is occurring in these early pathways. We also have a number of accessory organs which are present in the early GI tract, notably the salivary glands up here in the oral cavity, which release their secretions into the mouth. We have the teeth, which are technically part of the digestive system and are very complicated, and we're going to neglect them. And finally, the tongue, which is a large muscle here, which is involved in swallowing and mechanical breakdown. And so I just really want to highlight that this early GI tract is really primarily involved in conditioning food for absorption. So chemical and mechanical digestion and not so much absorption. So we're going to start our discussion with the oral cavity and the related accessory organs. Let's start with the salivary glands in chemical digestion, because we kind of covered that at the start of the lecture. So there are three salivary glands in the mouth. There's the parotid salivary gland, kind of in the cheek region. We have our submandibular salivary gland, which is where the name suggests, and also the sublingual salivary gland, which again is under the tongue, like the name would suggest. So salivary glands kind of have two different functions. So the first one is to produce saliva, and saliva is a specialized secretion which is produced by these serous cells within primarily the parotid and submandibular glands. And specifically, the enzyme that they're producing, or one of the enzymes they're producing, is an amylase, which begins chemical digestion. So an amylase is a enzyme that breaks down sugar, specifically starch, which is a polysaccharide. And starches are polysaccharides that are composed of glucose. So here we can see our starch molecule, and so our amylase is going to go and cut apart this starch, and specifically it breaks down the starch and chops it up into disaccharides, maltose, and oligosaccharides, dextrins, which are composed of 3 glucose molecules. So the salivary amylases aren't breaking it all the way down to the monomers, but they're breaking it into a lot of small pieces. And so if we actually look at these glands, just out of interest, you don't need to necessarily know this for the class, these have the compound tubulo-alveolar structure. So you can see they're kind of there's a large tube, and then they have these kind of sacs like the lungs, the alveoli. So they're sacs, except in this case, they're producing secretions in there. The other enzyme that is produced by the salivary glands is a lingual lipase, so easy to remember because it's lingual. And so this lipase actually catalyzes the hydrolysis of lipids. And so hydrolysis is going to be breaking of bonds by adding water. And so it's relatively intuitive, lipids, lipase, so it should be intuitive. So here's an example of what a lipase would do. Here we have a triglyceride right here. So it's a glycerol backbone connected to 3 fatty acid residues. And so our lipase can come in here and it can chop off these fatty acids and break our fat or our triglyceride into a glycerol, which is actually a sugar, and then 3 fatty acids. And so now this lipid can be more easily digested and processed further down. The other gland, the sublingual salivary gland, produces primarily mucus, and mucus serves an important role in lubrication. So imagine like if you have something really dry, it's going to be really hard to swallow. So it's kind of assists in this transport process, and it also serves to mix food. So the food is mixed with the mucus and lubricated, so it's kind of involved in mechanical breakdown. And so this is produced by mucus cells rather than serous cells, and then the mucus and the saliva mix together and mix with the food to form a bolus, which is ultimately swallowed. And it's just notable, if you wanted to differentiate these on the histological level, that the mucus glands have this compound tubular structure. You can see the difference is really in this bottom gland here, which rather than these sort of sac-like projections, it's more tube-like glandular ducts. So like I mentioned, saliva is a pretty complex mixture. It's mostly water, and it doesn't have so many solutes. So it actually has low osmotic pressure and fewer solutes than, say, in your blood. The solutes are numerous. For instance, protons, which is going to be acidic. So your mouth is slightly acidic, which is good for protection, but also a bunch of different electrolytes and solutes that will help kind of pull water with it, which will keep it hydrated. In addition, there's lots of different proteins, mucins, the enzymes we talked about, and also antibodies that are produced by your immune system. And so these are present in the saliva and obviously can help in protecting. And then some of these other things are involved in chemical digestion. The next organ is the teeth, and the teeth are very obviously involved in mechanical breakdown, and that's their role in digestion. And it is really important if we think about a really fibrous or large food, it's really important to get that mechanical breakdown in order to increase the surface area of the food for chemical digestion to take place. And you probably have experience of this. If you've ever not chewed your food very well, it comes out the other end undigested because it hasn't been broken down, and enzymes and stuff are not going to have enough time, and there's not going to be enough of them to get to the middle of a giant chunk of food. And so you really need to chew and break down that food in order to fully digest it. So this example of meat, which is composed of muscle fibers, which are also composed of smaller molecular fibers, and then here you can see the molecules of the fiber. So really breaking this down as small as possible so that these enzymes enzymes can get to work. And tooth anatomy is really interesting. And I'll also mention that enamel in particular is a really unique substance, and it's the hardest substance the body produces. And so there's lots of interesting things about the teeth, but it's just a little too much for the scope of this class, so we won't talk more about it. And I'm just going to apologize to any future dentists and mental hygienists, or dental hygienists, not mental hygienists. Next, we have the tongue. And the tongue, in terms of digestion, serves roles in swallowing and mechanical breakdown. And so swallowing or deglutition also involves the pharynx and the esophagus, and we'll talk about it in a moment. The tongue also has a lot of taste buds, which are going to be involved in controlling secretions from the salivary glands and also controlling sort of cephalic reactions or brain reactions to food and satiety and hunger. And so it does play a lot of other roles, but if we're thinking about digestion proper, it's really involved in transport and mechanical breakdown. So that naturally brings us to the pharynx, which we've already talked about in terms of the respiratory system, and the esophagus. So as a reminder, this pharynx is shared by the respiratory and digestive tracts. There's different parts of it, and hopefully your the nasal cavity is not involved in it, or your nasopharynx, and really it's these lower areas. And so the esophagus is simply a large muscular tube, and it branches off in the pharynx. So we have our trachea coming one way, and then our esophagus going down to the stomach. And so the esophagus, the pharynx, the tongue are all involved in this reflex of swallowing. And that's the primary function, is just transporting it to the next step. And so the swallowing reflex is really complex. It's actually coordinated movement across multiple things. So for instance, the tongue helps push the food back. But then we also have the epiglottis and uvula, which are also involved. So for instance, the uvula blocks the nose, so the food doesn't come up your nose, and the epiglottis blocks your trachea, so the food goes down into the esophagus proper. And then the pharynx and the esophagus are going to direct that food down and ultimately to the stomach. And here is the swallowing reflex, which we've already seen, but this black is the food that's coming in, and you can see as it's pushed down through the esophagus. Getting more into this idea of structure function at the histological level, the esophagus is really easy to distinguish from other parts of the digestive tract because it's very exposed to the external environment. And so its epithelial layer it reflects that exposure in that it's stratified squamous because it has to have multiple layers because stuff is going to come in and scrape against that. And so these cells tend to take a lot of punishment. And so here you can see an example of a histological section. This is the epithelial layer. As a reminder, those epithelial layers are very cell dense. And then underneath we have our papillary layer, which provides blood supply to that epithelial layer, which does not have its own blood vessels, and then lots of connective tissue. And here you can see what the esophagus looks like when it's kind of contracted and not full. It kind of folds up and gets these wrinkles in it. And here you can see a diagram of the structure. And so again, if we think about specializations in our alimentary canal, our esophagus is really going to be specialized for interacting with the external environment. It is worth noting that these epithelial layers are not keratinized like your skin, so they're not quite as protected as other parts, but they're also lined with mucus, which provides an additional layer of protection. So once food gets into the esophagus would push it down into the stomach, and the stomach is really where digestive processing starts in earnest. But as a reminder, the stomach is still not absorbing stuff. It's still involved in mechanical breakdown, in kind of storage and transport, but not absorption. So the main function of the stomach is actually for temporary food storage and also transport. And so you can survive without a stomach. There's nothing the stomach does that's absolutely essential to getting nutrients in your body, but it certainly helps to have some place to store food. And because it has such a capacity to stretch, it has these wrinkles or rugae or muscle folds in the inner lining, which reflect when the muscle is relaxed, it kind of wrinkles up. And then if you expand it, it can smooth out. So that reflects the fact that it has a very large capacity from 50 milliliters to 4 liters capacity. And some of you have noticed this in your minks that have dramatically different stomach volumes depending on how recently they ate before they were harvested. In addition, the muscle has another specialization at the gross anatomical level, which is that it has a third muscular layer. So remember the GI tract, the alimentary canal, all of the layers have all of the parts of the alimentary canal have these two layers, the circular muscular layer and the longitudinal muscle layer. The stomach has this additional oblique muscle layer. So you can see the orientation of the fibers is kind of diagonal. And so that oblique layer gives the stomach an extra range of motion. And so it has this additional churning motions. Think of like a butter churn, and it does exactly what you would think. It helps mix the food that's in the stomach. So the food's hanging in there, it's sitting in the stomach, and the stomach mixes it with gastric secretions. And these specialized movements are termed mixing waves. I'll also note that different parts of the stomach serve different or play more or less roles in some of the processes we're talking about, but I'm not going to dive into it in this lecture. But I do want to just highlight, if we look at our stomach and we take a cross section and look at the tissue level of our our layers of our alimentary canal, we can see our inner mucosal layer, which is interacting with the lumen, and then our submucosal layer with the blood vessels. In our muscular layer, we can see this bottom muscular layer, the outermost muscular layer is our oblique layer. And so that's what's generating the mixing wave. And so that's one of the easiest ways to distinguish muscle or stomach from other parts of the alimentary canal is if you can see these three muscular layers. Here you can see 1, 2, 3 layers of muscle. So the stomach is involved in both transport and mixing, so transport and mechanical breakdown, but it is also involved in sort of more molecular level interactions and digestive processes through the production of gastric secretions. So it produces two main secretions: alkaline mucus, which is really to protect the stomach wall. So this is mucus, it lines the cellular layers, and it's composed largely of water, and mucin and bicarbonate, which gives it its alkaline or its alkalinity, which helps buffer the tissues from the very acidic interior of the stomach, which contains the gastric juices mixed with the food. And when those two things are mixed together, they form chyme. And so these gastric juices are composed of acid, HCl, and obviously water, and it also contains enzymes and intrinsic factor. And so note that I've color-coded these by function. The acid serves a role in mechanical breakdown, specifically denaturing proteins. The enzymes act to chemically digest compounds in the stomach, and then intrinsic factor is important for absorbing vitamin B12, although it's not directly absorbed in the stomach. So let's look at the cells that compose the stomach lining, starting with the outermost layer, which is composed of the surface epithelium. So these surface epithelial cells contain many mucus cells, and these are what are producing that alkaline mucus. And so it kind of lines the stomach lining and is sitting on top of these gastric pits. And so this alkaline mucus prevents the stomach acid from damaging the cells. And you can see that in this diagram here. This stomach acid full of protons, H+, is interacting with the different food particles within the stomach proper, and it has a very low pH, indicating its high acidity. And then we have this layer of alkaline mucus, which has a high bicarbonate content, and this bicarbonate will bind to those protons and neutralize them and convert into water and CO2. And so you can see that this bicarbonate is actively being secreted from these mucus cells along with the rest of the mucus secretion. And so by the time we actually get to the cell layer of the stomach and the lining of the stomach, the pH is basically neutral. And so these cells aren't getting damaged by the highly acidic conditions in the stomach. And obviously, if the stomach mucus is compromised, for instance, if you're eating too many acids, you can actually overwhelm the mucus, and then you get something like an ulcer where that acid actually does cause problems, and it starts denaturing proteins and interfering with the lipids that form the stomach lining, and then can cause serious physiological problems. But basically, this bicarbonate is being sucked in from the blood and released to the mucus, while these protons are effectively being neutralized via this bicarbonate reaction. The next deeper layer, as we go deeper from our surface, our gastric pit proper, and our surface epithelium deeper into the gastric gland itself. So this is the gland or our duct right here. We have these mucus neck cells. And so these cells are also producing mucus, although and they are histologically differentiable from the surface epithelial mucus cells, although their function is not entirely known or understood why they're distinct populations. But I just wanted to mention that they're there. At these earlier layers of the gland, in addition to the mucus neck cells, we have a zoom in right here for this deeper part of the gland. We have this additional cell type, which are the parietal cells. So these parietal cells right here, which have this very high surface area morphology reflecting their importance in their role in producing secretions and getting that into this duct, produce hydrochloric acid or HCl, which is important for mechanical breakdown of proteins. So again, denaturing or unfolding those proteins and also kind of helping to break down lipids. So these denature proteins, they're also involved in activating this enzyme pepsin, which we'll talk about in a moment, and helps in breaking down cell walls and killing bacteria, although there are some bacteria that like acidic conditions. So it's kind of a mixed bag. But these are the parietal cells, which are colored in purple, and you can see they're all in the mucus neck. And so if you're wondering, so how did these secretions actually get into the stomach if this mucus should neutralize it? And so remember, we have this muscular layer here in the mucosa. So you can imagine these parietal cells kind of filling up the gland with the HCl and stuff, and then when and so that there's going to be rhythmic contractions, which is going to effectively squeeze the secretion out of here, past the mucus into the stomach, and then it's going to relax, and then those secretions are going to be produced. So that's all coordinated with the production of the secretions, which is another reason why it's important that the gut kind of communicate via autocrines and paracrines in the local neighborhood. Just want to mention that the protons or the acid that is produced by these parietal cells is actually produced from carbonic acid, which is what we've talked about a lot. And so that's synthesized as a reminder via carbonic anhydrase. So carbon dioxide and water are reacted to form carbonic acid. And so that carbonic acid, here we have our CO2 and H2O, our carbonic acid, and then that dissociates into bicarbonate, which is useful for mucus production, and then also protons, which can be used to bind with chloride to form HCl. And so effectively, what these parietal cells are doing is they're stripping off these protons from the bicarbonate or the carbonic acid, and they're releasing it and secreting it along with chloride from the blood plasma. And so what we actually observe is this chloride shift where there's a lot of chloride flowing out of the bloodstream into these parietal cells right by the stomach because it's going with this proton to provide neutral charge and generate the acidic conditions of the gastric juices in the lumen of the stomach. Finally, these parietal cells also produce the substance intrinsic factor, which is important and necessary for absorption of vitamin B12, but that's not actually absorbed until it reaches the small intestine. The next cells in the very deep part of the gastric glands are the chief cells. So these chief cells are specialized to release enzymes or zymogens, which are enzyme precursors that will actually do the chemical digestion in the lumen of the stomach. So the two enzymes are pepsinogen, which in the stomach lumen is activated and converted into pepsin, which is a protease that breaks down proteins. And then it also produces lipases, which digest approximately 15% of the lipids. So as a reminder, these enzymes are actually chopping and breaking these atomic bonds to break down the polymer into dimers and oligomers. I want to in particular mention how this pepsinogen works. So pepsinogen is released from these chief cells and then secreted. So here we have our pepsinogen molecule right here. So this is a protein right here. And once it's in the stomach lumen in the acidic conditions, the HCl that's released by the parietal cells interacts with pepsin, which is already in the stomach, and pepsin is the actual protease. And so these two act to cleave and cut off this peptide from our pepsinogen, which yields both pepsin and this free peptide. And now this pepsin is free to start breaking down proteins. And this is important because it makes sure that our protease isn't active within the gland itself, so it's not breaking down or damaging your cells itself. It's only activated when inside the stomach where it's acidic. And as a reminder, this pepsinogen is called a zymogen, which is an inactive precursor to an enzyme, and so it actually requires something, for instance, cleavage via pepsin in acidic conditions in order to activate into its enzymatic form, which is catalytically active. The final cell type at the very deep part of the glands are these enteroendocrine cells, which, as their name suggests, are involved in producing hormones and other signaling molecules like paracrines. So those paracrines act to coordinate stomach and intestinal activity. And specifically, I want to just go over the basic phases of secretions within the stomach. And so it all starts with the cephalic phase. And so cephalic refers to your head, so think brain. And so the cephalic phase begins or stimulates the secretion of things, of substances within the stomach. So it activates these glands. And so this happens kind of when you're eating or when you're aware. And so it effectively prepares your stomach for the arrival of food, and it's controlled by the parasympathetic division of the ANS, which, as a reminder, is interacting with the brain via the vagal nerve. And so here you can see kind of how it works. There's sight or sound of food or the smell. And so your brain effectively tells your gut, okay, get ready, we're going to eat soon via the vagus nerve, and that stimulates the secretion. All these diagrams are also going to have the things that inhibit it. So in case if you're, for instance, if you're depressed or you don't have appetite, that's also going to go through this pathway, but it's going to reduce secretion rather than increasing it. The next phase is the gastric phase, which, as suggested, or as the name suggests, occurs with events in the stomach. And specifically, as the stomach fills up and distends and stretches, that activates short reflexes in the stomach that figure to trigger both more secretions, because now there's stuff in the stomach that needs to be broken down, and starts mixing and churning within the stomach. And so this is also controlled by the parasympathetic division of the autonomic nervous system, but also via local signals in the enteric nervous system proper. And so there's things that can inhibit this. For instance, if your stomach is really acidic, then effectively it will sense that and shut itself down so you don't damage your own stomach lining. Finally, the intestinal phase is the last phase, which, as it suggests, is when the food is being moved from the stomach into the intestines. And so this is triggered as pH decreases. So as the food is mixed up in the chyme, which is what the food in the gastric juice mixture is called, and as the intestines start to distend, as food is moving into there. Into the intestines, which is actually happening through the pylorus and happens in sort of little spurts from the end of the stomach. So this is the pylorus. There's a sphincter here, which controls the release of the chyme from the stomach into the intestine, because you don't want too much going on in there at once. And so this is going to then trigger secretions from the small intestine, which is going to start breaking down the food further and absorbing it, but also local reflexes in the intestines and also through the autonomic nervous system. And it's going to inhibit gastric secretion because the stuff is mixed up and ready to start being digested. And here you can see the different things that are triggering this activity. So in summary, in today's microlectures, I talked about the structure-function relationships in the early part of the digestive tract and the GI tract, specifically from the oral cavity to the stomach. We went over the primary role of these areas is in starting chemical digestion and mechanical breakdown, and then finally transporting the food to the intestines, which is where the absorption will actually occur. And that's what we'll talk about in the next microlecture. Finally, we talked a little bit about how this activity is coordinated via hormones in the autonomic nervous system, which allows the stomach and the intestines and the rest of the digestive system to be in sync. So food is being digested and absorbed when it's available and being absorbed and broken down efficiently.